Well, thank you very much. You'll notice, those of you who've heard me talk before, that I went up with notes. I think I haven't done that since my bar mitzvah speech, which was 57 years ago. But it's so important to me, this conference and this wonderful day, that I wanted to make sure that I managed to say everything that I wanted to say when I was preparing the talk. I'm not sure if I'll manage. It's pretty hard to look at notes and look at the slides and look at the audience. So we'll see how it goes. I want to thank you, all the people, Boris and everybody else, for your very kind words during the day. It meant a great deal to me. It was a wonderful feeling and brought back memories in some cases, and gave me an appetite for more. This isn't my retirement party by any means. And if it's been such fun for the past 40 years, why not have a few more if, with the help of God, I have the brains upstairs and the health downstairs in order to do it. Well, thank you very much for that, and thank you for the wonderful talks we've heard today. I think I'll start with a story. A man goes into a pet shop, and he wants to buy a budgie, a tuki in Hebrew. So he looks around and he sees a nice-looking budgie on a perch. And he says to the seller, "How much is that budgie?" And the seller says, "Well, that's $1,000. Is that $1,000 for a budgie? That can't be." And he says, "Well, you know, that budgie speaks 5 languages, including ancient Greek." So he says, "Okay." And then he sees another one, and he says, "Well, what about that one?" So he says, "Well, the seller says that budgie is actually $5,000. And he said, "Well, what does it do?" And he said, "Well, that budgie can actually do advanced math, including calculus." And now he's really impressed. And he looks at more budgies, and every time the price goes up and the extraordinary expertise and talents of these budgies increases. Till eventually he sees a budgie sitting right up at the top all by himself. And he says to the seller, "Well, what about that budgie?" "Well, that budgie is $100,000. The man is totally floored. He said, "How on earth can a budgie be $100,000? What does that budgie do?" And the man says, "The truth is, I have absolutely no idea what he does. But the others give him a lot of respect, and they call him Professor." Well, I've been a professor for about 30 years, and so I think I'll try and tell you a little bit of what I've been doing all this time. But before that, a few words of thanks. First of all, of course, to Tsiona. Where is she? For her help and her support at every stage of my career with this conference, and for 48 wonderful years together. To Yoav for chairing this conference. Yoav was my mentee, and today he's a leader in child psychiatry in Israel and chairman of the Israel Society for Biological Psychiatry. To Harold Sackheim, Julia Lucinia, and Ofer Ragid for coming from the US and Canada to be speakers. Harold and Julia are dear friends. Harold told you from how long we've been friends, and Julia and I have been friends for quite a few years too. Ofer was my mentee, and I followed his career with pride since then. He knows some stories which fortunately he either forgot or chose not to tell. I want to thank Renana Eitan and Amit Lotan for the excellent talks. Both are my former mentees. Renana has returned from a postdoc in the States to direct research at Kfar Shaul at Eitanim hospitals, and to the great delight of her mother, I persuaded her to bring her return even earlier, and so I think her parents will be eternally grateful to me for that, as I am grateful to her for the wonderful talk she gave. And of course, Amit, who came back from his postdoc and is now a PI, a principal investigator in the lab with his own projects, hopefully his own very large grants in the future to keep the lab budget flowing. And thank you to both for your talks. I want to thank my brother, Dr. Leonard Lerer. Where is Len? There he is at the back as usual, bashful. Thank you for coming from Chicago to participate. It's really great to have you here. In absentia, I want to thank Dina Ben Yehuda for her kind words, Elliot Gershon for his words this morning, Tzvi Fischl and Igor Barosh, for what they said during the opening session. A lot of thanks, but I've got a lot to be grateful for. I want to thank my dear friend and long-standing collaborator, Fabio Macciardi, who came all the way from Irvine, California. With a name like that, he should be living in Milan, where he did when we started our collaboration, but good scientists get taken to the United States, and so did he. Fabio, it's great to have you back in Israel again. Thank you for coming. I want to thank Dick Epstein, who's here from Singapore. I'll talk a little bit more about Dick a bit later. Anna Alkalai, my former PhD student, now a postdoc in David Goldstein's lab in New York. And Eliyahu Dremenkov from Bratislava. Where's Eliyahu? Ah, there he is, yeah. Thank you all for coming. I want to thank Ashley Bush, Amit's mentor from Australia. It's like, like in-laws. When you have children who get married, then you have to deal with the in-laws. But I think I'm pretty happy about dealing with Ashley, and I look forward to that. And I want to thank all of you, my mentees, my students, my colleagues, collaborators, friends, family. A lot of you came from far. I thank those that came from the United States, but I think getting to Jerusalem from Tel Aviv early in the morning is almost as hard. Thank you all. So that's it. And if I forgot anybody, I'm really upset. And it was certainly not intentional. So now you know why I brought notes for my speech. Moving on to the talk, just a few disclosures. I'm advisor to a few biotech companies, and we hold a few patents. None of the things that I'll talk about today are relevant, except the last one, which is the deadenase 3 inhibitors, which is a line of research that we're pursuing in the lab. And I will talk a bit about that, but until anybody is persuaded to buy these drugs, it's going to take at least 10 years, so I don't think it could be considered advertising. So what am I going to talk about? This is the list. It's quite an ambitious list of things. It's quite eclectic. It's not a history of biological psychiatry for the past 40 years at all. It's very ambitious. I don't know if I'll finish it, but it doesn't matter, because whatever's important you can read in the literature, and whatever isn't important doesn't matter anyway. And so I'll just go along and get as far as I can. I do want to say that I've worked with a lot of people. It's been a great privilege. I've worked with a lot of people. I've had a lot of mentees, students, psychiatrists working with me. I'm not going to be able to mention everybody nor everybody's work. And what I've chosen is really quite eclectic, more just to give you a flavor of what different things I've been interested in over the years. And perhaps I've been interested in too many things. Yoav showed a slide of me winning the 400 meters at our school athletics competition. Maybe if I'd stayed in athletics and not played cricket as well as rugby, as well as other sorts of things, I would have been a good athlete, not just a jogger like I am today. But I have this tendency. I like things that are interesting. I like things that are challenging. I can't resist ideas, especially when young people that work with me bring them to me. So I get tempted, and I get dragged along, and I enjoy every minute of it. But perhaps too many things, who knows? I've still got a few years. Maybe I'll just concentrate on one thing from now on. So this is Groote Schuur Hospital in Cape Town. This is where I went to medical school. This is where I did my clinical training. This is where the first heart transplant was performed by Chris Barnard while I was a medical student. The cartoon at the bottom was done by a classmate of mine, David Westerman, and I think it's meant to both portray the fact that I was already observant then, but also the fact that I loved internal medicine. I had the most fantastic clinical education. I wish that Israeli students could have the kind of clinical teachers that I had there. I think when I got to Tel Aviv Shomer for my internship, I could auscultate heart murmurs better than the chairman of cardiology. And that wasn't because I was the best. That was because we did it over and over and over again until we got it right. As a result of the great clinical, I was always interested in psychiatry from a very young age, but as a result of the very intensive and excellent clinical education we got, I spent a while in internal medicine until I realized that psychiatry was, in fact, for me. This is Yaakov Avni, Zichrono Livracha, my first clinical tutor in psychiatry. Boris, I was so touched when you said that you remember my teaching you the clinical examination, because it was from Yaakov that I learned how to examine patients. Perhaps the pasuk, Moshe kibel Torah misinai, Maserar Yoshua, Yoshua leskenim, Moses got the Torah on Mount Sinai, etc., and he passed it down. But I think about 80 or 90% of what I know about how to examine a patient, I examined from Yaakov. I learned from Yaakov. And in addition, he also introduced me to the connection between depression and the thyroid gland, which I'll talk about a little. This is a patient, not one of my patients, taken from a textbook, but the patient I had was called Helen, and she was a patient with what they then called hebephrenic schizophrenia. She was my first patient in psychiatry, in the Department of Psychiatry in Hadassah. And I think that was when my interest in biological psychiatry was born, because psychoanalytic theories of schizophrenia were quite prominent at the time. And I, this cheeky young fellow from South Africa, looked at this patient. I said, it's not possible. This lady has to have a disease of the brain. There's no way that the way in which her mother treated her, and I was 24 years old at the time, there's no way in which her mother treated her could cause her to have this severity of illness. The last picture there is of Herman van Praag. Herman van Praag, many of you, the older ones, certainly have heard of him. Herman came to Hadassah in 1974. He was a very active researcher seeking the role of serotonin in psychiatric disorders. He did a lot of very interesting studies. And he came to Hadassah in 1974. And he gave us a course on biological psychiatry and psychopharmacology. For me, it was like Moses had come down from Mount Sinai and laid the Ten Commandments before me, laid the whole Torah before me. It was a fantastic course, and I was totally entranced. Of course, he spoke a lot about serotonin and so on. I wrote to Herman. He's now 96. He lives in Maastricht in Holland, and still writes a lot about Judaism and psychiatry. And he wrote me a really lovely letter wishing us well with this conference. Well, some of you, not many of you, will know what this picture is. Eliezer Pearl, where are you? Yeah, Eliezer knows this picture because we were students together in the psychotherapy course of the Psychoanalytic Institute at the Israel Institute for Psychoanalysis on Disraeli Street. And it was really an excellent course. We had fantastic teachers. The one I remember most of all is Eric Gumbel. Eric Gumbel was an orthodox, rigid psychoanalyst of German extraction or Austrian. But he knew how to teach. And whatever I know about psychotherapy and psychoanalysis, I learned from Dr. Gumbel. But I had problems with psychoanalysis. I see you're very smiling at the back. I had my difficulties with psychoanalysis. And in 1979, I wrote a paper. I was still a resident in the Israel Annals of Psychiatry, in which I explained why I could not live with psychoanalysis. Essentially, what bothered me was that they had an explanation for everything, and most times the explanation was based on resistance, and usually my resistance. So I wrote here, and I honestly tell you that I read Popper's article after this about why he rejects psychoanalysis. Psychoanalytically oriented therapy is a long and time-consuming process. Neither its endpoint nor its goals are universally defined. Too often, both seem arbitrary and malleable. This is what I wrote in 1979. But I will say I keep the certificate in my office very prominently displayed, and I must say that my experience in psychoanalysis and at the Institute had an enormous effect on my clinical career. I think that I learned a great deal there, and I don't regret being part of that course. Well, I really started in biological psychiatry in 1977. when I moved from Hadassah to Ezrat Nashim. And I moved in order to work with these two guys, Chaim Bellmaker, who apologized for not being able to be here today. He's giving a keynote lecture in Norway, I think. And Dick Epstein, who's there. Where is he? He was here. With a hat. How could I miss him? I went there because I wanted to work with these guys. They were doing translational research 30, 40 years before anybody had ever heard of or invented the term translational research. It was an absolutely fantastic experience. Everybody who was there grew up to be an important figure in Israeli psychiatry and in research as well. Avi and Ronit Weitzman were there. Ehud Klein was there, Yossi Zohar was there, and there was a fantastic atmosphere. One of the things I vividly remember is that Chaim used to give us research seminars in which he used to talk about things of interest. I remember vividly the seminar on the Danish schizophrenia adoption study, where my interest in genetics was born. And what also was rather amusing is that Chaim used to eat every single day for lunch during the seminar when he gave it as well. He used to eat tinned gefilte fish and matzah. And the funniest thing was to see the cat on the windowsill next to the window and waiting for the empty gefilte fish tin, which Chaim would put outside for the cat to lick. Anyway, I learned a lot there from Dick and from Chaim. I learned how to design experiments. I learned how to analyze data. I learned how to work with animals, with mice and with rats. And I even learned how to measure serum lithium levels with a flame photometer, which for somebody with two left hands like me is a pretty good achievement. And amazingly, Dick went on to be the PhD supervisor of my son Elad more than 25 years later. So what did I do there besides watch Chaim eat gefilte fish? This is the first experiment that I did in psychiatry, the first full-scale experiment. What did we do? At the time, there was enormous interest in the role of beta-adrenergic mediated adenylate cyclase in antidepressant action. And there was a very prominent paper published in Nature by Vetulani and Salsa. And so we set out to look for proof in humans that this finding was indeed valid and correct. And here we see what we did is we administered salbutamol to patients for several weeks, patients with depression, and then we infused salbutamol IV and looked at the plasma cyclic AMP response, which Dick measured. And what we showed was that as the treatment progresses and as the depression improves, the plasma cyclic AMP response to salbutamol is significantly blunted with time, matching the findings reported by Vetulani and Salsa. This was translational psychiatry, translational research, and I couldn't stop reading this paper. I used to read this paper every day, at least twice, after it came out. But then I suppose that is what one does when one publishes one's really first empirical paper. Well, moving on, that's also when I started to do research on ECT. I'd been fascinated by ECT, as Harold said this morning, since I was a resident. And it fascinated me, as Dina, the dean, said, because it worked. It was so amazingly effective. And I became very interested in the mechanism of action. Very early on, I published a review with Chaim, and a book which I edited together with Rich Weiner, which was really the first book on the basic mechanisms of ECT. And Harold mentioned this morning the anti-Parkinsonian effect of ECT. And this was one of the early studies we did, published in Neuropharmacology in 1981, where we showed that administration of chronic electroconvulsive shock to rats induces or increases dopamine-mediated behavioral responses. And this fits extremely well with the anti-Parkinsonian effects of ECT. Then came Detroit, 1982 to 1984. People said to me, why on earth are you going to Detroit? It's freezing cold. They do make cars there, but most of the cars are ragged and rusted from the snow, and the place is in total pandemonium and going downhill every day. I went there because of Sam Gershon. Sam was an icon in biological psychiatry, still is, thank God. He's 96 or 90-something, I think, Julio. Don't know. Anyway, he's certainly not 70. And he lives in Miami today. And Sam, for me, Sam was then the guy that introduced lithium to the United States, and he had worked with John Cade, the legendary John Cade, who had discovered lithium in Australia. And I really wanted to work with Sam. As we were about to go to Detroit, Tsiona, myself, and our 3 children, the Lebanon War broke out, the first Lebanon War. And I'd by then done my army service, so I spent a month in Lebanon in the war. This was really not nice. And people were shooting at me, and I don't think that they were playing games. I saw a lot of soldiers with combat stress reaction, and that's when my interest in PTSD was born. Afterwards, I didn't pursue it because I moved to Hadassah, and Arik Shalev was there doing PTSD research. But to this day, I do a clinic once every 2 weeks at Hadassah, and about 50% of the patients I see have chronic PTSD. So we went off to Detroit, and Sam was a fantastic mentor because he was a facilitator. He didn't tell you what to do. He gave you the resources. He guided you, he watched you, he checked on you, but you did what you wanted. And I did what I wanted. One of the things I did there was the first double-blind, randomized controlled trial of carbamazepine versus lithium in mania, in which we showed equivalent effects. And one of the interesting things, which I think even I have forgotten, is that there are specific patients who are helped more by carbamazepine than by lithium. I continued my work on the basic mechanisms of ECT. Harold referred this morning to the work on cholinergic receptors, and for this I received the Bennett Award in 1984 of the Society for Biological Psychiatry. One of the people who helped me a great deal with this work was the late Mike Stanley. Mike was a Jersey boy, tough, and had served as a helicopter pilot in Vietnam. He didn't like a lot of people. There were a lot of people he didn't like, but fortunately I was in the other group. Unfortunately, Mike died at a very young age, and I miss him still. So that was Detroit. We were there for 2 years, and I came back with my family and went back to Ezrat Nashim. There started a very, very productive period with Baruch Shapira, who was then a resident, my research collaborator, today became a professor, and now is working in the Ra'anana area. And we did a lot of work on ECT. We had 2 consecutive NIH grants to study optimization of ECT mechanisms. Harold kindly mentioned these studies. One was in the American Journal, the other in the British Journal of Psychiatry. The issue, the important point here was that I think we showed not only that giving the treatment slower allows you to achieve equal efficacy to giving the treatment faster after a little longer time, but it has very important implications for the way in which, for the neurobiology of ECT, for the way in which the therapeutic action of ECT is built. And I don't have time to go into this right now, but I think this is the cardinal importance of this work. We also looked at the effect of caffeine to enhance the ECT seizure. Harold tells us this morning that, or recently, that the seizure isn't important. I have to write a letter to the American Journal of Psychiatry regarding that and the paper. And of course, we did a lot of neuroendocrine challenges studies. Neuroendocrine challenge studies are something that my heart bleeds for. Why? Because they were so elegant, they were so translational, they were so clever, and they looked so good, and they yielded such interesting results. And then one day, people invented the PET. And in order to know what receptors are doing in the brain, all you have to do is bind them. So who on earth needs to give a drug which then releases a hormone, then measure the hormone? So things change, life goes on, and you have to adapt. In 1990, I moved back to Hadassah, and I was recruited there by Atara Kaplan-Denur, who, Zichrona Livracha, who recruited me soon after she had recruited Arik Shalev from the IDF. Atara was a true icon of academic medicine. She valued the importance of research and realized that for research to be done effectively, the most precious commodity was time. Not money, time. She made sure that I had enough time in which to do my research, and made sure that the residents on the ward were involved with me. And several of them are here today, and most, if not all of them, are professors somewhere or other. And Atara, I owe her a great deal, and I truly wish that the heads of departments and the heads of psychiatric hospitals will realize this, that the importance of academic psychiatry lies in giving people not only the encouragement to do research, but the time to do it. And if they have the time, they'll find the money. But if they don't have the time, they'll never find the money, and they'll never do research. And research will be number of papers published and not number of truly important discoveries made. Two people joined me on my move from Ezrat Nashim to Hadassah. Sadly, they're no longer with us. One is Michael Newman, mentioned this morning by Elliot. Michael was an outstanding scientist, a fantastic biochemist. He was not my mentee, he was my peer. Michael had an NIH grant in his own right, very, very rare for someone outside of the United States to have an NIH grant for basic research, but he had one. And the other person was Rachel Alexander. Rachel was a social worker. She gave up a sure job at Ezrat Nashim to move to Hadassah on soft money. And she trained the whole cohort of people who afterwards recruited all our samples for genetic studies. And I think a lot of the people here, several of the people here, I see Anna there and Amichai, worked with the DNA and the data from the samples that were collected either by Rachel or by people like Kyra Sana, then called Kyra Kanyas, who actually collected the samples. Well, we attracted a lot of people, not only clinicians, but also PhD students. I hadn't had PhD students until that time. I'd worked primarily with psychiatrists. And every year we have a barbecue at the home of Yoav and Odi Cohen's house. And every year the numbers grow and people keep coming back. And it was a fantastic time over the years. And I'll tell you a little bit in a moment or two about what we did at Hadassah, what we're doing at Hadassah. But I do want to mention one more thing, and that is Hadassah Brain Labs. Hadassah Brain Labs is a organization which we set up based on the research capacity of our own laboratory and the research capacity of the Department of Neurology Laboratory headed by Professor Chana Rosenman, who I see sitting up there. And it's been a fantastic collaboration. We got really excellent grants from the Ministry of Science. We provide services to other researchers in Israel. We provide some services to companies as well. We learn a lot of techniques, and we also make some money, which is critical for keeping our research going. And finally, two people that are, finally for the slide, two people to whom I owe a very great deal. The one is Dr. Tzuri Lifshitz. Tzuri was my PhD student and then postdoc.
And one of equal, the whole team was there for tuition.
Everybody will get their mention, Tzuri. There are primum inter pares. And Tzuri is the lab manager today. We've done a lot of research together. I rely on Tzuri. Tzuri's our foreign minister, because somebody mentioned, I think the dean mentioned this morning, that I have a tendency a little bit to being impetuous, and whenever somebody needs to be spoken to in a hospital that requires a cool head and somebody who doesn't get irritated, we send Tzuri. And of course, Amit attests I try to adopt Sam Gershon's facilitation policy, but Amit tests this policy to the utmost, as you could probably have guessed from his talk this morning. He also tests, but he's a fountain of ideas and activities, and it's great to have him back. Well, what have we been doing? I'll talk about a few areas of interest. The one is thyroid. I've always been interested in the ability of thyroid hormone to potentiate antidepressant action. As I told you, Yaakov Avni told me about it. By the way, Yaakov had returned from a fellowship in the lab of no less than Joe Schildkraut, the man who wrote the famous article on the catecholamine hypothesis. And we know that that people with hypothyroidism tend to be more depressed, tend to have a higher incidence of depression, and people with depression have a higher incidence of thyroid disorders. And it's also very well known that giving an antidepressant to patients with giving thyroid hormone to people who are not responding to antidepressants can improve their response. So I took this over the years and kept coming back to it. This is work done with Eitan Gur and Michael Newman using microdialysis, where we showed that thyroid hormone enhances the release of serotonin, and another study showed that it aggregates with the effect of imipramine. This is work that Suri did, very elegant work in the JPET in 2011, where we showed a clear dose-response effect for the antidepressant effect of thyroid hormone. And this is work done with Rena Cooper, published in the then Archives of General Psychiatry, now JAMA Psychiatry, where we showed the clinical effects. Well, I was very interested, but already several years ago was born in me the desire, the wish, to develop a compounds that could be used to enhance thyroid hormone effects, but without the adverse effects of long-term thyroid hormone treatment. So what did we do? We tried several different approaches, Turi and I, and then we hit upon an idea. We know that deiodinase 3 breaks down T3, and that deiodinase 3 is specific to the brain. And so if you give a patient, if you can inhibit deiodinase 3, you're going to inhibit it in the brain, somewhat in the skin, but not in the heart and not in tissues where its adverse effects could be important. So we teamed up with an Indian biochemist, Indian pharmacologist and physical chemist and pharmaceutical chemist named Govindasamy Mukesh in Bangalore. I found him after an intensive literature search, looking for somebody who was interested in thyroid and synthesizing a new compound. And he synthesized a series of DIO3 inhibitors for us. And we've been working with them. We've got newer data, but this is one of our earlier studies showing that one of these compounds has clear antidepressant effects in mice. And we have more data from recent studies which support this. But one of the things that happened, and this is why I talk about time. You have to be able to read. You have to be able to read papers that are not critical for what you want to do right now. And I read about DIO3. Anything comes out, I'll take a look at the abstract, and if it's really interesting, I'll read it. And then I realized from my reading that DIO3 is significantly increased in certain cancers, in cancer of the colon, in ovarian cancer. And it struck me that DIO3 inhibitors could inhibit the growth of tumors. Why is DIO3 inhibited? Because thyroid hormone causes differentiation, promotes differentiation. And if you can, and the cancer cell does not want differentiation. The cancer cells want to be undifferentiated. And so if you, the theory I had was that if you can inhibit deiodinase 3, then you could perhaps inhibit cancer growth. And I'm not a cancer researcher by any means, so I teamed up with Dr. Osnat Ashur Fabian. This is one slide from a series of projects that she's done, that we've done in collaboration. And it shows clearly that in ovarian cancer, high-grade ovarian cancer, you can see that the cells treated with IOP, you can see the significant difference in the density of the growth of the cancer cells. Since then, we've teamed up with FutureX, which is a biotech accelerator funded by several large pharmaceutical companies. We've gotten some very helpful grants from the Ministry of Economics. And right now, we're approaching the stage where we're going to go to the next stage of development. Well, that's about thyroid. A lot has been said about genetics. I was attracted to genetics because I believed that I needed to find some route to understand the biology of psychiatric disorders. I felt a bit of an interloper. There were people like Eliot who'd been doing elegant genetics for years and years. And Ted Reich, who was one of the leaders in the field, asked me if I know advanced mathematics. I said, not as far as I know. And he said, well, forget about genetics then, it's not for you. But I have chutzpah. I'm born in South Africa, but I've been in Israeli for a long time, so I decided nevertheless to get involved. And we started to collect families, because that was what one did at the time. And one of the family samples that we collected that was most impressive, we collected several different samples, was from the Taibe area of the Mishulash. And this kind of sample from large families, there's no increased incidence of schizophrenia in the Mishulash, in the triangle, but the families are very large, and they were all treated in a central clinic run by Dr. Adnan Hamdan. I'm not sure if he's here. He said he may come. No, he's not here. And so we collected these family samples, and it's a long story, and it took a lot of years, and I'm certainly not going into all of it. But working with Fabio Macciardi, and this was the basis of very long and very important collaboration, and Doron Lancet, who I see sitting over there. Why are you taking notes, Doron? You know all this.
It's because you speak so eloquently. I need to internalize it.
Thank you, Doron. Anyway, we worked with Doron at the Weizmann Institute, and with several other people, including Daniela Aman, who was our joint PhD student, who's now in Australia. And we eventually identified a locus on chromosome 6q called AHI1, Abelson Helper Integration Site 1 gene. It's a gene which is very important in neurodevelopment. These are the papers that we published, several of them over the years, going from the initial linkage study to the discovery of the gene. And remember, this was before GWASs. This was a time when if you wanted to do SNPs, you had to do them individually, and doing 100 SNPs across a chromosome took about the same time as sequencing a genome today. And this was done in Doron's lab, and Fabio worked with us intensively on analyzing the data. Of course, we were absolutely delighted when other people replicated us. This is not a gene which is part of the 108 loci, which Eliot erroneously attributed to me. I mean, I definitely contributed to the paper, me and about 750 other people. But it's not one of those loci, AHI1. But it definitely has a very important role, we believe, in schizophrenia, and probably in a subsample of schizophrenia patients. So I started out working with Epstein and Bellmaker on mice and rats at Izrat Nashim. I went back to them a few years ago, this time transgenic mice, and we obtained mice which have reduced expression of the AHI1 gene. And we worked very intensively, the whole group, on phenotyping them. Gili Wolf, Hagar Ben-Ari, in addition, of course, to Tsuri and Amit, worked with us on this, and here's a Western blot which shows that these mice do indeed have underexpression of AHI1. This is a, I'm not sure how to make it work. Anyway, it doesn't matter. Oh. Yeah. So these are mice in an elevated plus maze, and any normal mouse will avoid the the open areas, and these mice, in this particular segment, they don't, but AHI1 mice actually spend much more time, as you can see from the bar here, in the open segment of the elevated plasmase, indicating that they have reduced perception of the danger of being in this area. And we have a lot of other tests which support this. And being a psychiatrist, of course, my first interpretation was, I never lost my psychoanalytic training, was that they have some kind of impaired reality testing, that they really don't know that they're not supposed to be there. They're also resilient to stress. Here we see that after being submitted to chronic stress, all the mice except the AHI1 knockout have reduced sucrose preference. And of course, they have significant abnormalities of brain connectivity, as shown on functional MRI. All this is in 2 papers published in Molecular Psychiatry with Amita's first author, and one that is in Translational Psychiatry right now with Gilly as first author. Well, I'm not going to talk much about pharmacogenetics. The time is moving on. But Lerer Greenbaum is here, I think. I saw him at lunch. There he is. We did a lot of work on pharmacogenetics, a lot of work on the genetics of smoking, but there really isn't time for that, because I do want to end on time. One more thing I can't resist telling you about, and that is my foray back to Freud. I really enjoyed his article, Mourning and Melancholia. And the issue of loss and depression always stayed with me. And when Ofer Agid was working with me, we designed a study to look at the question of whether there really is early parental loss in patients with psychiatric disorders, depression, but also other disorders. And we simply did an empirical study in which we collected a large number of patients and controls and inquired as to the existence or non-existence of early parental loss. That's when I really got to know Giulio. I wrote to him, I said, this isn't molecular, Dr. Lucinio, but it's very important. Will you review it? Well, he reviewed it and published it, and it became a very highly cited paper because we clearly showed that loss is associated with both depression and schizophrenia, and in addition, what we show, and bipolar disorder. But we also showed clearly that it was not the death of a parent that was important, but the separation. In other words, traumatic separation of one kind or another. Divorce, abandonment, parent going to prison for an extended period. Loss of that type was associated with depression and other disorders. And then there was another study done by Laura Canetti, in which we collaborated, on Israeli adolescents, which actually supported that observation. So, what do I think about the future? I agree that brain stimulation is extraordinarily important. I look forward to all the developments, and great that Renana is back, and I think a lot's going to come there. I think there's another area that is important, and that is the interaction of environment and genotype. I think that we've not found any major genes that contribute to susceptibility to psychiatric disorders. I am fairly certain we're not going to find, except in very rare cases. And I think that the interaction of genotype, of gene and environment, is where it's going to happen. And that's the reason I showed the work done on early parental loss in order to bolster this point. I think for those people in the audience who are at the beginning of their scientific careers, I'll say go there if you're not going to brain stimulation. Go to gene-environment interaction, because that is where it's going to be at. Well, one other thing I did was the IJNP. It took up 10 years of my life. I founded the journal, as they say in Hebrew, Yesh Mi'ayin, at the request of the CINP. We rose to in less than 10 years to an impact factor close to 5, and I had a great time editing the journal. After 10 years, I decided enough is enough, and I handed over the reins to Alan Fraser, who was one of my field editors. Irena Luna, whom some of you met outside, all of you met outside when you picked up your badges, she was the office manager of the journal for a full 10 years. And I don't think we would have gotten to where we got without her contribution. So that's it. That is the story. Thank you for listening. A few more comments. I see I stopped looking at my notes, so I have to now go back and see about the end. Yeah, so I've told you 40 years in what I hope was 40 minutes. I hope that the younger people here take home the message that there are few things in the world as interesting as biological psychiatry. Research in this field is immensely challenging, but hugely rewarding. I hope the department and hospital heads who are here will take home the message that clinical excellence and academic excellence are two sides of the same coin. The dean said this morning, and this is my true belief, not everybody has to be a researcher. You can be a fantastic doctor without being a researcher. You should have a little bit of a taste so you know what it's about. And not every researcher has to be a doctor. Most of the research in the world is done by non-MDs. But there is an entity which I think is absolutely critical in any medical school, in any university hospital, and that is the physician scientist, the one who is both a clinician and a researcher. But that doesn't come by making empty promises or making statements which have no cover in reality. It has to be based on support, commitment, and time. Time, as I've said over and over, is the crucial issue here. And I got that time. I got that time from Atara Kaplan. I got that time from Arik Shalev when he took over as the head of the department. And I tried to give it of that time to the people that I work with. And of course, one of the other, besides the intellectual rewards of doing research, there's of course the rewards of the people you meet. You meet fantastic people all over the world. You get to make a lot of friends. It helps to be able to drink beer, which I do, and wine even more so, which I do even more so. But you get to meet a lot of people. It's enriching, and it's really very, very meaningful. I want to thank, I've mentioned all the people in my group except the youngest of them, Anna Dona, not Anna Alkali, who joined us recently and is doing great work. We're a small, tightly knit group. Everybody works twice as hard as normal people, so that's why there are only 6 with me, but we generate data for about 12. And I'd like to thank our collaborators. A lot of people have put their money and behind their faith in our work, and I'm grateful to have received that. So somebody preempted my slide. It was Yoav, but if anybody asks me what is the message that I take home after 40 years, this is it. For me, mentorship has been the most important thing of all. To meet everybody here today, many others I was in contact with, I sought out every single person that I had done research with and published a paper with. There were many other people that had done research, but I had to stop somewhere. So that was the cutoff, doing research and publishing at least one paper. And I had fantastic responses. Not everybody could make it today. I've received a lot of thanks for what I've done for them. I deeply appreciate the thanks, but I want to let you into a very important secret. And that is that I received a lot more than I was given, and I've been enriched a lot more than I've enriched others. Thank you. From the applause, you'd think I scored a goal in the World Cup.